Hello, and welcome back to another episode of the Curiosity Monkey podcast. Now, as living things, we are constantly faced with our own mortality, being we will die at some point, and that risk of death increases as we age. In fact, it doesn't increase linearly, but more in an exponential fashion. So as we get older, our risk of death increases more and more. In fact, aging is the biggest risk factor for death in older humans. But aging isn't consistent across the animal kingdom. In fact, today's episode, we're going to be talking about the immortal jellyfish, as it's called, and how it manages to bypass death. So the immortal jellyfish is also known as its actual name, Turritopsis dohrnii, and it's a small jellyfish that lives worldwide, typically more in temperate to tropical waters, so warmer waters. And it's one of the few animals that is thought to be able to theoretically live forever and not die. So whereas most animals get older and they age, they tend to die in pretty much every case. This animal is thought to be able to continue living indefinitely. So the jellyfish of the Turritopsis dohrnii is bell-shaped with a kind of a diameter of 4.5 millimeters, so quite small, kind of maybe the size of your finger, and there's a large red stomach, which is easily visible from the outside of the jellyfish. Now, this jellyfish was discovered in 1883 when scientists collected these things called polyps. Now, I'll explain a bit more about what polyps actually are a bit later, but polyps are essentially what jellyfish are kind of born from. So they kept these polyps and they monitored them until it's called the medusae stage forms, which the medusae is like the actual jellyfish part of the life cycle. And again, we'll go through this in a bit. So they thought these jellyfish would mature and then spawn some larvae. But they found actually when they left them for a while that they turned into new polyps. So they went from the polyp stage and these produced jellyfish. And then these jellyfish somehow managed to go back to the polyp stage. So this has really intrigued scientists. It's kind of like when a caterpillar can turn into a butterfly, and then the butterfly turns back into the caterpillar form. So in a second, we'll go over the mechanism of how these jellyfish manage to revert between jellyfish form and the polyp form. But these jellyfish are found kind of all over the world. It's thought that they originated in the Mediterranean Sea and then got transported through ships because they're so resilient and can survive quite harsh conditions. They can survive in the ballast tanks of ships. So to maintain buoyancy, ships kind of have these ballast tanks which they can fill with water, and so it's thought that the jellyfish kind of got in there and have been transported all across the world. So what is the life cycle of this jellyfish? Well, swarms of these jellyfish release eggs and sperm, so the females release eggs and the males release sperm, and these eggs are then fertilized and can, after a while, develop into planular larvae. And these larvae are able to settle on the seafloor or wherever they manage to settle, and eventually they develop into things called polyps. So you may be wondering, what is a polyp? Well, polyps are kind of cylindrical-looking creatures with one end anchored to the seafloor or some kind of solid surface. The other end houses a mouth with this ring of large extending tentacles. And these tentacles are organs which can basically act like sensors, but they can also be for the capture of food. So polyps extend their tentacles, particularly at night, and they kind of uncoil and can hold on to prey and pierce them and poison them, paralyzing them to kill them. And they prey on all sorts of different things, including fish larvae. And then the muscle within these tentacles kind of contracts and pulls in the prey to the mouth. So the polyp is thought to be quite a simple structure, and it's thought that these creatures have not really changed that much for about 500 million years. And it's also thought that they predate the dinosaurs by at least a few 100 million years. So they're very old, and in fact jellyfish haven't really changed that much over this time. Jellyfish predate dinosaurs, and it's important to mention here that many other types of jellyfish form through the formation of polyps and then are released as jellyfish as well. So it's not just the immortal jellyfish that forms like this. And it's kind of hard to study them in the long term because they don't really have bones. So fossil records typically are boned creatures. So the soft tissues just kind of disintegrate over time, and it's hard to actually see these creatures such a long time ago. So it tends to be quite rare that you find a fossil of a soft-bodied creature from such long ago. But the polyps are basically what these jellyfish are produced from. So as the polyp is anchored to the sea floor, so it comes from the larvae, which are from fertilized eggs, and these planular larvae then settle on the floor, and then they form these polyps, which are anchored to the floor. And then on the other end of the polyp, they kind of produce these stacks of medusae, which are the jellyfish form of this creature. And then eventually, when the jellyfish are ready, they then bud off from these polyps and then continue their life in a free swimming form in the ocean. Eventually, they will become sexually mature after a few weeks. So both the single planula that the polyp colony formed from, all of the polyps, and all of the medusae which came from these polyps are genetically identical. So they've got the same genes, they've got the same DNA within them. So when the polyps release the medusae form, or the jellyfish form, the jellyfish can then grow and they prey on different things. So for any reason the jellyfish is exposed to significant environmental stress, or is injured through maybe someone tried to eat it, or it's getting old, it's getting sick, diseased, then it can remarkably revert back to the polyp stage, forming a new polyp colony. So like before, the analogy typically used to describe this is from going from a caterpillar in a cocoon to a butterfly, and from the butterfly form back to the cocoon stage. So the medusa of the immortal jelly is the only known form of jellyfish or animal to have the ability to transform back to the polyp stage. So all stages of this jellyfish seem to be able to do this, whether they're really young, just kind of being released, or they're really old, fully mature, they seem to be able to transform back into the polyp stage, but under specific conditions. So as I said, if they're getting old, they're injured from something trying to eat it, you've got sudden changes in environment which just don't favour it. Environmental conditions like changes in temperature or changes in the salinity of the water, tissue damage, all these things can lead to the transformation of the medusa form, jellyfish form, from its jellyfish form back to its polyp stage. So this begins with deterioration of the bell-like structure. So like when you typically see a jellyfish, you have the bell, the head of the jellyfish, and then it was tentacles. So this bell, the kind of hydrostatic tissue that's inside the bell, and the tentacles, they all kind of blob or transform into a blob-like structure called a cyst on the seafloor. And to do this, the cells kind of go from a differentiated form, where each cell has a given function, back into a less differentiated form. And we'll get onto this in a bit. But the cyst is basically a small capsule that kind of holds all of the jellyfish's cells. The cyst then, after a while, can develop back into polyps. So this occurs after about 2 days. However, it's been seen that some jellyfish just skip the cyst stage. They go straight from jellyfish medusae form back into the polyp form. So once the polyp form has been achieved again, then the polyps can grow and develop a stack of medusae just like before, which can then bud off and swim away as jellies. And these jellyfish are all genetically identical. So you've got the single planula, which kind of fell to the seafloor and developed a polyp colony. And then these polyps released the medusae, the jellyfish. And then a few of these jellyfish perhaps became injured and reverted back into a cyst stage, and then back into a polyp stage on the seafloor. All of these stages are genetically identical, which then produce more jellyfish. And the process that allows this to occur is called transdifferentiation. So the cells in the polyp stage are different to those in the jellyfish stage, meaning the cells have to transform between different types, which is quite remarkable. So if you think in your body, you've got loads of cells. Each cell has a specific function, what the cell is meant to do. And this is determined by kind of what genes are turned on and off. So in your body, pretty much all your cells are genetically identical. You share the same DNA. All the cells share the same DNA. But what the cells do with this DNA is determined by something called epigenetics. So what genes are turned on and what genes are turned off. And this controls the cell type, so like the structure of it, the size of it, what its function is, so what metabolites does it produce, what does the cell do, what's the whole point of it. This is all determined by the control of the genetics within the cell. So the jellyfish cells will turn on and off different genes so it can revert from jellyfish cells with jellyfish identity to polyp cells. Can perform a different structure, its shape changes, its size changes. The whole activity within the cell changes just by turning on and off a few different genes within the cells. So it's this process, transdifferentiation, that allows cells from one type to go back and revert into a different type, completely different type of cell. So transdifferentiation is just the process in which a mature specialized cell, so a cell with a given role and function, can change into another. And does this without entering a pluripotent state, which is basically when you develop a stem cell that can differentiate into many, many, many different cells. So this allows the jellyfish cells to be able to transform into polyp cells, and then the polyp cells to be able to transform into jellyfish cells. So sorry if I'm just going over this a bit too much, but I have to make this clear because this is the very cool thing about this jellyfish, its ability to do this, because in humans we have a very limited ability for this to occur. So this allows the jellyfish to become biologically immortal because they can theoretically continue this forever. Now, this has kind of only been observed in laboratory studies, but it's been observed countless times, and there's no reason why you wouldn't see this in the wild under unfavorable conditions to this organism. So what is the significance to science? Well, most cells in humans specialize to fulfill a particular function, as what I was just saying. So you've got, say the epithelial cell in the lung that facilitates diffusion of oxygen into the blood, or neurons that transmit electrical signals. They all have the same DNA, but the control of that DNA is different. So epigenetics, where you're turning on and off different genes. So this turning on and off of genes will control what genes are expressed and therefore what the cell actually is and what it does. So this is called differentiation. It changes dramatically what the cell's size is, what its shape is, what it does, the metabolic activity, how it responds to different signals within the body. And in dividing cells, there are multiple levels of cell potency. And this is the ability to differentiate into different cell types. So if you have a greater potency, it means you have a larger number of cells that can be derived from that cell. Now, most cells cannot differentiate into other cell types once they're specialized. So if you have a given cell, it's going to stay as that cell. It's not going to become any other type of cell. So it will continue as that cell until it dies. So these jellyfish could hold important information in regenerative medicine to replace lost cells, damaged cells. Now, humans do have some ability to regenerate and restore tissues. So, for example, red blood cells actually produce from a certain type of stem cell from the bone marrow that differentiates, produces red blood cells. So that's an example of going from an undifferentiated state where that stem cell could become different types of cells to a differentiated state where a stem cell is differentiated, it's become more specialized to form, say, a red blood cell. Now that red blood cell is fixed as a red blood cell. Another example is in the liver. Liver cells, or the liver can regenerate quite remarkably after significant damage. So in the initial case with the red blood cell, this was through activation of different stem cells. So the restoration of different tissues can occur through, say, the activation of stem cells to divide and specialize into given cells to replace damaged or lost cells, or the proliferation of already existing cells. Some vertebrate species can regenerate very well. So, like if you chop off the limb, it could regrow it, for instance. So this is quite remarkable, considering a human, if you chopped off their arm, it would not grow back. But maybe through learning about this type of medicine, we'll be able to develop therapies that can help replenish lost or damaged cells. Now, to do this practically, you need a good understanding of the mechanisms that allow you to take one cell and transform it into another cell, as in the process of transdifferentiation. So the process of transdifferentiation in the immortal jellyfish occurs in 2 steps. So first, the jellyfish cells de-differentiate. So they step from a stage of differentiation, where each cell is, say, terminally differentiated, where it's in a specialized form, and it can kind of revert into a less differentiated form where the cells are less specialized. Now the cell has de-differentiated, it has the opportunity to re-differentiate into many different cell types. So you have the jellyfish cells, they step back into a less differentiated form. And then this less differentiated form re-differentiates into a new cell type, completely different. Say, a polyp cell. Now, both the polyp and the jellyfish are made of a large variety of different cell types, so this de-differentiated cell has to be able to re-differentiate into many different cell types, so it can cover all the cells required to build, say, a polyp or a jellyfish. So this process happens in newts as well, and so newts are quite well known, so if you chop its kind of leg off, it will just regrow it. Now this transdifferentiation can also happen in the lens cells, so if the newt lens is damaged, then pigmented epithelial cells from other areas of the eye can de-differentiate, and this kind of causes them to lose their pigment, changes the shape of the cell, and then the cell can re-differentiate into lens cells, which then can heal and replace damaged tissue. And this importantly allows the organism to regain functionality. So there has been some quite cool research on using transdifferentiation to change cells of one type in a body to a different type. For example, converting cells from the liver into pancreatic beta cells, these are the cells in the pancreas that produce insulin. So the reason for doing this is trying to regenerate these cells from people with type 1 diabetes, where they've been destroyed, can allow functionality back and regain of control of blood sugar levels. So by doing this in mice, they managed to actually treat the hypoglycemia. So when the blood sugar comes too high after eating sugar, the pancreas would typically release insulin from the beta cells in the islets of Langerhans, and this would reduce the glucose level. So hypoglycemia is when blood sugar levels are higher than they should be, and insulin should be released. So in diabetic mice, type 1 diabetic mice, they have hypoglycemia after eating because they cannot control the blood sugar. But after treatment of these cells, which came from the liver and were converted into pancreatic cells, they were able to regain control of their blood sugar and reduced the hypoglycemia, so reduced the high blood sugar. There is also a lot of hope that we'll be able to do this on humans by getting liver cells again, and they were able to transform them into, to transdifferentiate into human beta cells. So basically the benefits of this is, again, gaining functionality of the pancreas to produce insulin to control blood sugar levels. So there's all sorts of different gene therapies that could treat a huge variety of different conditions. And this would be pretty cool. So being able to cure people from type 1 diabetes, for example, then they'd no longer have to monitor blood sugar levels so strongly, and they wouldn't have to inject insulin. This would be a very large breakthrough in science. So to recap for this episode, because we strayed a little bit away from the immortal jellyfish and into potential applications of the science that can be uncovered from it. So the jellyfish release eggs and sperm, these fertilize, you get planular larvae, which then settle to the seafloor and grow into these things called polyps. The polyps then produce these stacks of medusae, which are then released as jellyfish. Now, if the conditions, say, for the jellyfish are unfavorable, or they age, or they become damaged, then they can revert back into the polyp stage. And this is through the process of transdifferentiation, where cells of one type are able to go and revert into a cell of another type. Because ultimately the cells between the jellyfish form and the polyp form are very different, however the DNA is the same. And theoretically they can do this forever and ever. And so the polyps can then release more jellyfish, and then those jellyfish can revert back to the polyp stage, and it can continue. But this doesn't mean they cannot die. They are biologically immortal, which means they can kind of bypass aging, but they can still be predated, like eaten up, or just die before they manage to revert back into their polyp form. So it doesn't mean they'll live forever, but it does give us very interesting and cool insight on how we could potentially use transdifferentiation for our human medicine to develop new and exciting regenerative therapies. And at the end of the day, are they really immortal? Sure, their DNA might live on forever, as they are genetically identical, but it's almost like when I'm close to death, I go into a little ball and release loads of tiny little versions of myself, all genetically identical, but are they really the same as me? So that is about all for this episode of the Curiosity Monkey podcast. I hope you enjoyed it and found some of this content interesting. If you'd like to suggest anything that we cover in a future episode, then please don't hesitate to contact at thecuriositymonkey1@gmail.com, or you can message on Instagram. We have that at thecuriositymonkey. And so yeah, that's about it for this episode, and see you in the next one.